Thanks, Dean Norris, for coming over and celebrating with us our department's 25th year anniversary. Okay, so we're starting the last plenary of today, and it's going to be on biomedical engineering transforming healthcare in our communities. And we have four amazing faculty here. They have very long awards and success to talk about. Unfortunately, I will not be able to talk about all of that, so just give a brief summary. So we have Professor Randy Bartels here, who's an investigator of the Margaret Institute for Research and a professor of BME, who recently joined our department, and we are very happy to have him with us. And among many awards that he has received are Adolph Lomb Medal, Sloan Research Fellowship, ONR Young Investigator, and many more. Thanks for being here, Andy. I have Professor Chris Saha. He is a professor of biomedical engineering, and at UW-Madison, his lab works on gene editing and cell engineering of human cells found in retina, central nervous system, liver, and blood. And he also has many, many awards, and among them, some of the important ones are Next Generation Council for the Allen Institute for Brain Science, NARSAT Young Investigator Award, Career, and many others. And so thanks, Chris, for being here. And we have Kip, and Professor Kip Ludwig is a co-director of the Wisconsin Institute for Translational Neuroengineering and leads the Ludwig laboratory within the institute, focusing on developing advanced neuromodulation therapies that use minimally invasive techniques to address nervous system dysfunction and precisely deliver molecules to targeted areas. And before joining us, Kip was a program director at NIH, and he has also a stellar career, also very active in the entrepreneurship domain. He's the co-founder of Neuronov. How do you come up with these names, Kip?
It's a very bad pun.
Okay, and NeuroWorks, and several other companies. And we also have, last but not least, Professor Colin Wissenberg, who is the Jane and John Mandula Assistant Professor in the BME department. And her lab, the Cardiovascular Biomechanics Lab, has been awarded grants from the NSF, American Heart Association, and the Children's Heart Foundation, and also the WARF on campus. So thank you so much for being here. I will give the floor. Who's starting? Randy.
I guess. I don't know.
Go ahead.
We have a presentation. I don't know. Did something? Are there slides? I think there are slides, right? What's that? I had slides, yeah. Well, I guess, so while the slides are getting sorted, I'll just, there they are? Oh, hey, there we are. Here's our magical button. Okay. All right, well, it's my pleasure to be here. So I've got into Madison a couple of months ago. I've been back and forth for quite some time, and kind of the move here is really kind of, in my view, a time to kind of re-energize and go off on a new adventure. I had no plans of moving, and was coming out for a colloquium in October of 2022, and I got an email from Paul a few days before I came out that said, oh, by the way, Mortgage is doing a senior hire, and I assume you're not movable. And my response to that is, of course I'm not movable. That sounds terrible. And then I came out and I visited and really kind of fell in love with Madison, and in particular the people here and the opportunities for collaboration. So we've heard lots and lots about how collaborative this environment is, and that really, I'm really looking forward to digging into and doing that, because that certainly seems to be true. And of course, Kevin is playing matchmaker to everyone on campus, so thank you, Kevin. It's a real pleasure to be here, and like many of the people you've heard from already, my background is very different than where I am now in biomedical engineering. So I started in strong field atomic physics, built big lasers, shot them at atoms and molecules, ripped out electrons, controlled quantum dynamics, and at some point I decided I couldn't figure out how to explain why I was doing that to other people, let alone myself. And so I decided to switch, and I pivoted to microscopy, in part because we had done a lot of work in building lasers, and going into nonlinear microscopy was kind of a natural switch. And so my perspective on microscopy really is that it's a field that has a long history and lots of applications, obviously, in biomedical engineering and lots of applications in biological sciences. And so there are a number of errors that we might be able to identify. For example, early on, just trying to improve magnification. So just seeing smaller things with magnification era. And then there was a sort of a huge range of contrasts that evolved, many of them phase contrasts, say fluorescence, and it came probably really in the 80s with advanced labels and fluorescent tags, transgenic fluorophores. And I argue that we're now kind of in a phase where we're really pushing into a quantitative aspect of biology. So echoing what we heard a bit earlier, lots and lots of data. So and so this couples with computational imaging, data science, et cetera. And so what I'm really interested in doing is being able to look at chemistry and the dynamics inside of live organisms. And a lot of what we're doing here, so much like Melissa Scala's lab and Paul's and Kevin's, is looking at label-free imaging. So we want to be able to get away from using labels, but labels are powerful, use them when we can. But this is a bit like, thank you, Kip. Kip has found Waldo, because Kip knows that Waldo has some unique characteristics. So Waldo has a striped shirt and jeans and a hat, and so if we kind of put together a collection of characteristics, we can go and find Waldo, and that's kind of what we want to do with imaging. It's to take various sorts of optical properties and in terms of the light interaction with proteins and tissues, and be able to use that to identify specific, say, tissues or molecules or structures and dynamics without having to introduce labels that can perturb the dynamics a bit. So some of the various characteristics of interest are, we can get various shapes of cells, even down to organelles. We can look at things like second harmonic generation, which was pioneered by Paul and is really pushed by Paul, and analysis also by Kevin here. So there's a lot of innovation here historically in Madison. And one of the areas I've been working on is trying to do very sensitive vibrational spectroscopy to learn about, to do chemical imaging without labels. And in the end, my entry into this area was all with nonlinear microscopy, because I had built lasers, short pulse high intensity lasers. And it turns out the idea here is you can do nice imaging because you localize the signal you generate into a very small spot. So this 2-photon image on the right here, you can see a small spot, or maybe you can't because the projector's not great, but there's a small spot where light's generated. And since it's generated at a single point, you can raster scan this thing around and build an image in 3D. And one thing we've been doing, so you can do this with various things. For example, second harmonic generation. Again, these are images from one of Paul's review papers. So it's really an area that he has pioneered, and you can look at things like certain types of collagen structures and muscles, and this gives you this intrinsic contrast where you don't have to introduce a label, and you can see these structures. You can analyze these structures looking at morphology. You can quantify these, and so the thing on the left is work from Kevin's group, where they quantify the relative alignment of these collagen fibers around tumors to grade tumors and determine how dangerous these are for metastatic potential. So there's a lot of information that's available in this. So one of the issues with, here's another one. So this was work from my lab, so a number of years ago, where we were looking at layers of retinal cells. So you can differentiate different layers of retinal cells with two types of nonlinear microscopy, third harmonic and two-photon fluorescent microscopy. But so you have all this capability to do imaging, but the ability to drive this into applications is somewhat limited by imaging speed. So one of the things we've been looking at is how we can speed up imaging and how we can go deeper inside of tissue and do this at high speed. And so the basic idea is to do holographic imaging, and the reason for that is you're mixing light in a way that gives you a signal boost, so you can image at higher speeds and across larger fields of view and do this rapidly. But the problem is, as you go through tissue, we all know that we can't see through tissue optically. This is one of the challenges with light-based imaging. And so there are, and part of the reason for that is light is scattered in various directions. It scrambles spatial information, prevents light from penetrating into the tissue. And some of that distortion can be seen in this example, showing distortions of waves as they go through. In this case, it's the tip of a C. elegans worm. And this sort of distortion prevents you from seeing detailed structures, say, inside of this worm. So one of the things we've been working on very recently is being able to computationally undo some of these distortions. So this is just a quick example, again, of collagen fibers. So on the left, we see an uncorrected image. And if we take the right set of data, process it, we can correct for the distortions and get intensity and phase images. And this phase information gives us new possibilities for characterizing samples and looking at, say, assessing disease pathology or diagnostics, as well as for assessing potentially treatments with therapies. We've extended this to third harmonic generation as well. So this is an example of looking at osteocytes in a developing bone tissue. And then another area of interest here is to do label-free histopathology. So an issue with histopathology is it's a long process. It can take 24, 48 hours to slice tissues, process them, and produce a histopathology slide. It turns out you can do chemical-based imaging to do this very, very rapidly. So we've been we've been developing ultrasensitive chemical imaging techniques to try to push the ability to do that, as well as look at the dynamics of other molecules inside of biological systems. And I think I have too many slides here. I'm probably out of time. So I'll just sort of leave you with kind of this is an example of the world's fastest nonlinear microscopy based on chemical imaging. And so this is a wide-ish field technique where we need to sort of scale the lasers in order to do this over a larger field of view. But this is kind of the first generation version of being able to push to higher speed as well as do some super resolution imaging with this. Well, so here's some of the images that come out of this. You can zoom in and qualitatively see that the images on the right are these cars images. We get higher spatial resolution, and these are the fastest wide field images of chemical imaging that have been produced. So with that, I will, I guess, pass this up to Chris, who I think is next. But I just want to say I'm thrilled to be here. We're slowly. What's that?
Let's see the next slide.
Oh, okay. I think I put too many slides in here, and then let's see. Do other things too. Blah 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 blah. And let's see. So I don't know who's next here. But at any rate, I think I was taking too much time. So I'm thrilled to be here. looking forward to collaborating with lots of people, and I'm just proud to be part of this incredible history of this department. I look forward to contributing lots of things. So with that, whoever is.
I think I'm listed next in order. Let's hope my slides come up. So I was given 5 to 7 minutes to represent neural engineering across campus. We have 40 investigators who do some aspect of neural engineering, who hopefully can stop that sound. They cross 14 departments, but I speak fast. However, if I have my slides, it would be great. Anyone? I didn't even know what I'm supposed to be hitting. There we go. That looks promising. All right. Does this advance? Oh, that's even better. All right, so I know Dr. Justin Williams talked a little bit about some neural engineering that's been done on campus earlier today, specifically a lot of his work with neural interfaces for the brain, predominantly for prosthetic uses. I'm going to talk about a different use that is actually the largest growing market in the medical device center, which is known as bioelectronic medicines. It's also known as neuromodulation devices or electroceuticals. But it's really a combination of a couple things. One, we're all kind of familiar with getting orally administered drugs. And orally administered drugs are very useful for certain conditions, but when you take them, you don't get the drug where you want it, when you want it. It has to be metabolized, it goes pretty much broadly to all your body, and if you want that drug just at your heart, it's waxing and waning as it is metabolized. Different people metabolize at different rates, and you're not keeping that drug constant where you want it, when you want it. This is a big problem for side effects, but it's also a big problem for habituation. Anytime you get a biomolecule which you have a receptor for, if you start getting an abundance of it, you get a downregulation of those receptors, and you start to adapt to it over time. So neural engineering is really the combination of two things. One, it's the idea that we have these electrical wires called nerves in your body that we can hijack. We can sense their electrical signal, and we can stimulate to induce electrical signals. What's really cool about them is they're actually drug delivery devices. They send an electrical signal down the wire, but at the end of the wire, at a synapse or end terminal, they actually release your body's own biomolecules, many of which are very similar to what you're taking orally. But the other really cool thing about them is your body has its own natural sensors for things like glucose, dopamine, pH, oxygen, and it encodes it with a biomolecular signal, but then it sends it back up these wires as an electrical signal. So by hacking these wires, we can not only deliver a drug just where we want it, when we want it, but we can monitor your own level of that drug and keep it constant throughout the day. So my mother doesn't like me when I call it hacking. She views that as too negative for neural engineering, so she likes me to call it wiretaps. So the other thing that this is a combination of is really the progression of technology. Raise your hand if you've heard of cardiac pacemaker. Most of the people here. So in this corner right here, do I get a laser? I don't have a laser. That's sad. I like lasers. This is the Hyman defibrillator. This came out in about the 1920s or 30s, and it was used to pace the heart. It's actually like the size of this table, and it has a hand crank to create electricity to go to wires to pace the heart at the time. But the progression of the technology follows Moore's law. It gets half the size, it gets twice as powerful and half the cost every 2 years. It's the doubling of Moore's law. This is the Zoll pacemaker in the 1960s. It's roughly about the size of this by this. They called it an implantable pacemaker, which makes me laugh because there's rotary dials in front, but in general, you'd wear it in your chest and the wires would go to the heart. This is what most of the people in the room probably think of when they think of cardiac pacemakers. It is an implantable battery that also has a little bit of onboard computing. You can wirelessly talk to it, and then we take a wire and we connect it to this battery in your chest. We snake it down the vasculature. We have an electrode in the ventricle, and it passes electricity to pace your heart. This is in 500,000 patients a year, and has been one of the most successful medical devices ever invented. This is not the current generation of cardiac pacemakers. This is. So Boston Sci, Abbott, and Medtronic all have their own versions of this. These are little miniaturized wireless pacemakers. They're about the size of your thumbnail. There is no implantable pulse generator in the chest. There is no wire going down. Instead, it has its ability to stimulate for 10 years. You can wirelessly talk to it, but it also has sensing capability. It can measure your EKG and when you're exercising, increase the amount of which is pacing your heart as well. It even has sensors such that if you're having a heart attack, it can send a wireless signal to your cell phone to call 911 as well. So now we're getting from these invasive ways to hijack the body, to teach the body to deliver its own drugs where and when you want it, to minimally invasive injectables that are getting to the point with work like Dr. Aviad Hai within the biomedical engineering community here, to be little motes that can go in through a needle and be injected. So an example of transforming medical technology, this is a company that I worked with, and this is actually a device I developed, and hopefully the video played. This is a 1 millimeter electrode that's placed on the carotid sinus to treat people with intractable hypertension, people who are unresponsive to drugs. And what you're doing is stimulating a nervous pathway to release neurochemicals at your heart and neurochemicals at your artery to slow down your heart and to open those arteries to reduce blood pressure. This is an example of a super responder, because I get to cherry pick my examples. If it plays, uh-oh. I get to lie about it. Can somebody hit play for me? As all I have is this. Bueller, raise your hand if you're listening to me in the distance, who's IT. Okay, there we go. Didn't know where IT was. So what you're going to see is a blood pressure trace of a subject. You're going to see the device turn on. The subject is blinded. Actually, could you start at the beginning very quick? It's a very quick video. The subject is blinded. I am not in this case. They're going to be, a blood pressure is about 170 over 90 or 100 if I'm measuring. So you see it in the right. You see a trace that's real time. The numbers are average. The device just went on. You see the blood pressure trace already going down. When it gets to the end, the numbers will update. Again, someone completely not responsive to drugs for hypertension. There's a timer on the lower right-hand sleeve you can barely see. 100 millimeters of mercury in about 2 minutes. It's hard to prescribe this to a placebo effect. This is now an FDA-approved device. The device off is not blinded because I believe I said something like, oh shit, pardon my language, because this was one of the first patients that we programmed, and it worked better than we were expecting. It goes back up 100 millimeters of mercury, all within 5 minutes. This is something that drugs are very hard to do orally administered, is have them turn on and off and titrate like this. So what's the power of that? Okay, now I need to get off of this screen and go to the next one. I don't have advanced control anymore. I got 5 to 7 minutes, man. Oh, that almost worked. Control. The next slide's probably the most important one. You can ignore most of what I say generally, but the next slide's kind of important. All right. There we go. So you may not realize it, but you're in the middle of what's called Medical Device Alley. There are, from Chicago to Minnesota is the Silicon Valley for this neurotechnology. There's over 100 companies in neurotechnology. There's over 1,000 clinical trials right now. And as an NIH program director, there weren't many new drugs approved while I was there. They were mostly generics and things like that. We've had these neurotechnology devices. You can almost not name something there wasn't an FDA approval for, whether it is hypertension, heart failure, overactive bladder, chronic pain, opioid withdrawal, ADHD, blindness, epilepsy. These have all gotten FDA approvals, and most of the companies are right near Madison. This is the place I wanted to be because if you want to transform health via the Wisconsin idea, this is the place to do it. So I can't go through all of these, but you can imagine this is complicated. This is why it spans 40 different investigators in 14 different departments, from basic neuroscience to physiology to clinical to mechanical engineering to electrical engineering to biomedical engineering. So this is why we created the Wisconsin Institute for Translational Neural Engineering, which is really to coalesce all this activity, but to do one more thing. Right now, we don't know our predictability of our animal models for neurohacking. We're doing a lot of animal work, but we don't know how well it predicts human success. And we wanted to create an institute that could do what we call parallel translation. We're not only trying to make things go fast forward to the clinic, but when they fail, understand if the animal models were predictive and if we could create better transforms like they have in drugs to predict our success rate as well. So this is a study where we're just using the great resources as really an agricultural college as well, across multiple animal models to understand the wiring diagrams from mouse to rat to canines to monkeys to pigs to humans. And not surprisingly, given this is the size of each, the dosing in each is very different. So a lot of clinical studies in my field have failed because they took a rodent dose and they thought it would work in humans. So as part of this accelerating, I believe Justin Williams talked about Neural One, which is a company that got uplisted on NASDAQ. We have CBRX is the company I just showed, which is also uplisted on NASDAQ. Neuronoff and NeuroWorks are two spinouts from Whitney that have started clinical trials with concepts that started on the bench in 2018, which shows how fast we can do this. But most importantly, we're connecting it to the educational system. When I left grad school, none of my teachers had ever translated anything to clinic, and mostly what I was taught, I had to unlearn because it was incorrect in terms of supply chains, insurance reimbursement, regulatory, how things are done. We have now several professors who are spinning out companies and actively doing this, and Dr. Wally Block will be teaching a medical device ecosystem in the spring, who also has, I believe, has 2 companies you're associated with. That's why it's so important for our BME faculty to be actively participating, because regulations change, where you do things change, the supply chain changes, and we can keep our students current. So thank you very much.
All right. Good afternoon. I'm Chris Saha, and I'm very excited to share what we've been up to. Also been really thankful to be here and hear about some wonderful stories over the last 25 years. They have these labels for professors as early stage, early career, mid-career, or just professor. And I think I came here in 2012, and I'm probably technically mid-department, since I've seen kind of the last 12 years or so of this wonderful department. And, you know, I got a better sense of time also from this morning, because ever since the pandemic, my sense of time has all been very compressed or expanded. I really don't know what's going on in terms of what happened before the pandemic in some ways, but you know, I think for my field of research, there was a kind of breakthrough and landmark moment around the founding of this department, which was the Human Genome Project and mapping the human genome. And so there was 3 billion letters that was mapped, and the question was, you know, what does this mean for patients, for really medicine? And that was a question that I was interested in, but really, I think we're at a really almost interesting landmark moment now, about 25 years later. So I came to Wisconsin here, just since everyone gives their origin stories. In 2012, I had degrees in chemical engineering, and I did a postdoc in biology at MIT. And at that point, I said I really wanted to work with stem cells. And he said, well, there's only a few places in the world, and arguably the best place is Madison, Wisconsin. And so I came here, and one of the things that was really eye-opening was the degree to which engineers and biologists and clinicians really sat around a table, talked science, critiqued each other's science, and came up with really interesting projects and solutions. So that was just a really important formative moment for me to come here. And since then, we've been working in this field of trying to write the genome within human cells. And it's based upon really the mapping that was done about 25 years ago. So, what's amazing now is that there are patient stories that I can convey. 3 of them are here: Victoria Gray, Emily Whitehead, and Michael Kalbrerer. Victoria Gray was one of the first patients with sickle cell disease to be treated with a gene-edited cell therapy. And this is a hematopoietic stem cell therapy that was just approved last December in the US and also in the EU. And you can read and hear about her stories of response. They're pretty remarkable and durable now many years after treatment. So that's an approved drug by Vertex. A clinician should be able to write down a prescription, and you can, if you need this drug, should be able to get it. Emily Whitehead is one of the first patients that was treated with a genetically engineered T-cell therapy, which are now being gene-edited as well. And there's also remarkable stories of her living a pretty full life now, more than a decade later. And then on the far left for you is Michael Kalbrerer, who has a rare inherited form of blindness called Leber's congenital amaurosis, and he was also treated, in this case, with an experimental genome editing drug in the back of the eye, and he had trouble seeing light, and he describes this moment at his cousin's wedding where he was able to see light for the first time. So these are pretty profound, life-changing types of interventions. And in many ways, they're all being advanced now by this technology that was discovered right when I got to Wisconsin in 2012 called CRISPR. Many of you probably heard of it, but for those who don't play with it on a daily basis, there's kind of 3 major components to CRISPR. There's this orange piece of a short RNA, about 100 bases or so, a protein, in this case called Cas9 nuclease in purple, and you could optionally have that blue nucleic acid that could be used as a template. So the RNA and the protein complex together, and once it's inside the cell of a nucleus, it searches the genome, really arguably all 3 billion letters at some frequency, and looks for a match to a sequence that matches that orange piece of RNA. If that happens, the protein makes 2 cuts in the DNA that you see, and the cell takes over. So there's endogenous DNA repair machinery that sense double-strand DNA breaks. And if this nucleic acid is present, it will be used as a template to write in new sequences where you made the break. So this gives you kind of a cursor to edit bases in the genome. And so this is being used by many in the US, and one of the beauties of NIH and federally funded work is that they fund a lot of collaboration across investigators, because we all have complementary sets of expertise. So this is one effort that I was fortunate to lead for the first 3 years of the effort, which is to make new types of ways of getting this machinery into different tissues and cells. There's groups that develop new editors to make more versatile types of edits in the genome, and also efforts to make little kind of micro tissues or tissue chips, things that would represent the brain or the eye or the kidney or liver, et cetera, whatever you're looking to edit. So 2 stories here of what we're trying to do with this, together with clinicians, biologists, and many people in this room, Bill Murphy, John Cruddy, Melissa Scala. They've all helped put this kind of team science effort together, where we're trying to get a first-in-human trial of a CRISPR therapy, very similar in some ways to what Michael Kalbrerer was treated with. But in this case, we're trying to deliver it without any type of viral elements. And the hope is that these rare forms of blindness, the ones that affect these channels in green here, Both dominant disorders that would, in this diagram would be red, could be either corrected in a recessive disease or in a dominant disease disrupted such that those cells get rescued function. These currents of potassium and chloride that normally would go through these proteins essentially would be rescued by doing these gene edits. And so this is an effort that spans 5 years and really partners across the coast to move this forward. And then my last slide here is to CRISPR cells outside the body, and this is also enabled by really long-term collaborations with folks in the Cancer Center and clinicians. Christian Capitini's in the room, and he works with many of us engineers. Takes time off from his busy schedule, treating patients to talk to us about how to make better cell therapy such that we can have more stories like Emily Whitehead, such that these T cells or NK cells could be genetically engineered to have better potency and efficacy. One of the more interesting strategies and challenges in the field is that the manufacturing of these cell therapies is actually limiting because it's made in a kind of bespoke way, one patient at a time. And we're really interested in making cell therapies that have higher durability and access and kind of a batch mode such that a clinician can just take a drug product off the shelf that could be used in all sorts of conditions, not only cancer, but more indications like autoimmune diseases, transplant scenarios, and the list goes on. So hopefully let's give you a snapshot of where we're going, and very excited to celebrate here. Thanks.
Sorry, I'm a little shorter than the other folks up here. So I think it's been a long afternoon, and I'm the last person who's going to talk to you before the next thing. So I just was hoping everyone would just take a nice deep breath and remember that we're going to be done in about 5 minutes. So hopefully I will be relatively quick. I'm the most junior professor on the panel, so I should have the least to say, right? Because I've been here the shortest time. So I'm Colleen Witzenberg. I'm the Jane R. and John G. Mandela Assistant Professor. To talk a little bit about my background, I started as a mechanical engineering PhD student and did my postdoctoral training at the University of Virginia. And that's really where I think I thought more about how my work was going to impact people, right? Up until that point, I had thought a lot about tissues. I'm a biomechanist, and I think about cardiovascular tissues, but I hadn't really thought that much about the people it was going to affect until that point. I'm sort of the opposite of most of our students. I'm really excited about the math and the science, and it wasn't really until later that I thought, oh, this can also impact people. And so one of the things that really brought me to the University of Wisconsin-Madison was this confluence of sort of three components, which is an excellent engineering school, a phenomenal medical school, and a fabulous veterinary school, because I knew what I wanted to do was think about how I could study biomechanics in the lab, in animal models, and then also how are we going to translate this to people, right? And so in order to do those things, you need those three components to facilitate your research. So that's really what I was excited about coming to the University of Wisconsin-Madison. Just to talk a little bit about some background here, when we think about the significance of cardiovascular disease burden within the United States, there are over 1 million heart attacks every year, right? So that's a huge number of people. We actually, as a community, have gotten really good at treating heart attacks. About 85% of people who have a heart attack survive. Unfortunately, that's not the end of the story, right? Those folks are then at risk for heart failure. We've got 6 million Americans living in the United States with heart failure. Many of those have their heart failure because of valve disease or aortic disease. And then there's this population of people that we don't talk about very much, and that's congenital heart defects. So despite the fact that we don't talk about them very much, 1% of the population is born with a congenital heart defect. 2 out of every 1,000 are born with a serious congenital heart defect. And this was one of the areas that I was really excited about because I thought that our science could be impactful. And so I just want to impress upon you that what's happened in the cardiovascular space is that we've had a lot of advances in intervention that involve surgery and devices that treat the acute. Right? We've gotten really good at treating heart attacks. We've gotten really good at treating patients who need a defibrillator. These sudden, acute, let's intervene now and save a life. What we see as our role in my lab is this question about what happens after that. Because unfortunately, what happens after that isn't that you go back to normal in many cases, but that now you're living with the consequences of this acute event. Right? You're living with looming heart failure for years and years and years. And so in our lab, we combine computational modeling approaches along with in vitro and in vivo data in order to make predictions. So I always talk about the computer models that are out there, these big, beautiful finite element models that folks have been using to make virtual surgeries, right? They're gonna decide how big a shunt should be or what angle it should be. But ultimately, those models only predict what's gonna happen the day of surgery. It sure would be nice if we could make models that predict into the future whether or not this surgery is going to have a long-term or chronic benefit for the patient. And the population for whom that is most important is our congenital heart patient pool. And so that's really been the passion of my lab, is thinking about these individuals, right? If we do the surgery differently when they're 6 months, will that mean a big difference in their life when they're 40 years old? And so I'm just going to talk a little bit about congenital heart defects, which is a defect that our lab is working on. We're working on 2 in particular. HLHS, which is hypoplastic left heart syndrome. It's the most severe defect that you can have. Those infants are born without a left ventricle. So for those in the room who've taken cardiovascular class, there are 4 chambers in the heart. Which one's the most important? Left ventricle, yes, that is the powerhouse, the pumping chamber. In the 80s, this was virtually fatal if individuals were born with this disorder. Through a series of very revolutionary surgical procedures, now most children with hypoplastic left heart syndrome will survive, and the rates of them making it to age 30 are at about 75%. But remember, survival is not long-term health, right? So how do we tip them from not just surviving into thriving? And the other story I like to tell is sort of on the opposite end of the spectrum, which is for a congenital heart defect called coarctation. This is when you have a narrowing in your aorta. Seems really simple, right? Go in, cut out that portion of aorta, stick in a stent, you're done. All the pediatric cardiologists in the 80s were like, we got this. These kids look normal, everything's great. And now they have a problem. This whole population is coming back to the clinic in their 40s, and they have hypertension as if they were 70. And we don't know why. And so this is where this idea around prediction is starting to come at, right? What is it that we are changing, and how is that impacting the growth and development of the heart and arteries over time? So I'm going to tout my first grad student's work, or my most senior grad student's work, I should say. So when we started, we had this computational model of adults, and we realized adults don't grow. At least they shouldn't. Our heart should not grow in an adult. But of course, in children, you have normal somatic development. So the first thing we had to do in our model was get it to grow, but to grow in a somatic way. And so she went out and she found a ton of literature data, and she went ahead and fit our model to do this. So you can see our model is predicting the change in mean arterial pressure for a normal infant from age 0 to 3. You'll notice mean arterial pressure jumps quite a bit in those first few weeks of life, and then stays pretty much stationary. For left ventricular parameters or measurements, the end diastolic volume is going to go up for the first 3 years of life. The model is nicely well within the literature data, and then she also is showing thickness there. So if volume is what is in the ventricle, thickness is the muscle, right, that you have. So then she went ahead and she simulated this aortic coarctation, right? So this is this stenosis of the aorta, and she showed that that increases blood pressure, as you would expect. But it also causes both a thickening and dilatation of the ventricle. And one of the big questions for coarctation is, do those things reverse when you fix it? And if they do reverse, do they go back to normal? And on top of this, we know there are also changes going on in the vasculature. And so part of the work in my group right now from one of our new students is to model how are the vessels responding to this change in terms of their material properties. Is the collagen and elastin that is starting in these vessels still there, depending on the pressure change? Sort of news, or maybe a flash forward, in animal models, that's not the case. And so one of the questions is, if you have this for a prolonged period of time, maybe you have permanent damage to your vessels that then doesn't affect your health until you hit your 40s. So with that, I will close, because I know we're all tired and ready for a break. But I wanted to at least acknowledge my wonderful group. So again, as a junior faculty, right, I just started my group in 2019, and so I wanted to show wonderful pictures of my great group. So the students, my graduate students, are Ashley Hebing, whose work I showed today, Daniel Pierce, Matt Culver, Kate Eberman, and Callan Kozitska. They do all the work. I'm just the person who helps with things. And then I also wanted to just say, one of the things that we've been really excited about our lab has gotten funding from the Children's Heart Foundation. And so just a note on this conversation around diversity and inclusion, one of the things the Children's Heart Foundation asked us to do is they asked us to come to one of their events and talk about our research. I was really nervous. I was like, I'm going to talk about my research in front of a bunch of people who are, being affected by this. Like, what are they going to think? And they were incredibly welcoming. And so now we've gone back year after year after year. We now even do a fun science activity with the kids at these events. And so I think one of the things that we have to think about when we're doing science is to get out of your comfort zone, right? It's easy to say they should come to me, they should come to campus, they should come to Expo. But I have to say, going to them has been one of the most rewarding experiences as a scientist and an engineer, and also one of the most informative. I was nervous about, I'm not making a device, I'm making a model. And the parents I talked to said, having some idea of what's going to happen in the future for my kid is invaluable. Right? And so that really bolstered my scientific motivation to go after this problem. So anyway, with that, thank you all for your attention. Hopefully we'll get a break.
Questions? People are really, yep, there's one. Go ahead.
So I talked a lot about clinical translations within the U.S. here. Are there any important challenges or areas for growth in branching out international translation of these sorts of medical advances?
Actually, can you hear me? So when I was in industry, we used to go to Europe first because you could get CE mark very quickly for medical devices with what's considered open label data that's not double-blinded or sham-controlled. Now the medical device regulations have changed in Europe, and it's trying not to use a swear word show. So that's how fast it changes, that we used to all go to Europe first to do kind of an open label trial that would serve as our phase 1, phase 2 trial in the United States, and then do the pivotal trial, phase 3 trial here. Now no one is going to Europe first because the regulations have changed so fast, and there's, especially for certain sub-devices of medical devices, we have no certainty of what is needed.
It's a really important question, and I think the field as a whole, at least in the gene therapy field, cell therapy field, is struggling with it. So there's a famous line by Jennifer Doudna, who won the Nobel Prize for CRISPR, saying that a $3 million drug, a $3 million cure is not a cure, meaning that if that sickle cell drug costs $3 million, there's a lot of people that cannot basically access it. And if you look at the burden of sickle cell disease, it's clearly not in the U.S. as the largest concentration of people in need is certainly in Africa, and secondly, is in also in India is the second most prevalent cases. So going to a place like India or Africa and saying I have this miracle drug, but it will cost you $3 million is not really attractive. So there's a lot of efforts to do this. One of the efforts here is our partnership with the Global Health Institute to explore different ways of manufacturing these cell and gene therapies. So Dr. Marcella Tabima and Dr. Sri Kumar Sengupta are here in the audience. They've established collaborations to think about what it would take to manufacture those CRISPR cell therapies out there. And it's a different set of infrastructure, different set of people, resources, and it's really interesting because, regulations are also different, but, it's not that they just let anything fly. They actually want to and get a lot of credit for going by the highest level of regulations like the ones here that the FDA mandates. So it's just a different approach, and I also think it's through like a public health regime rather than, a U.S.-based market distribution model. So lots going on there. And a similar type of story about rare diseases and gene therapy. That's also a similar set of challenges on access. Maybe doesn't cut across the same way in terms of the globe, but certainly within the U.S., there are rare disease patients, foundations, groups that are struggling to get effective therapies developed and also reimbursed. Lots of challenges there. I think it's one of these unique questions that academia and industry can come together around, and it's exciting.
Any other questions? Okay, if not, let's thank all of our speakers.